So welcome back. We are starting now with the NF1 session, and it's my pleasure and honor to introduce our keynote speaker, Professor Eric Legius. Professor Eric Legius is a clinical scientist, and he was the head of the Human Genetics Department of the University in Leuven, and also for many years the clinical director of the Center for Human Genetics of the University Hospital in Leuven. He made contributions towards the understanding of the molecular etiology of a number of NF1-related tumors, but also he contributed to the understanding of the mechanisms of microdeletions and also the development of MPNSDs. Professor Eric Legius is also the discoverer of the Legius syndrome, which shows similar features like Café spots as in NF1, and contributed to the revised diagnostic criteria of NF1 Legius syndrome and also the different types of schwannomatosis. And he's also a co-author of the tourist guidelines for NF1 related tumor surveillance and many, many more, but I don't want to keep you away from his talk anymore and hand over to you, Eric. The floor is yours. Thank you so much for being with us.
Thank you very much for this very nice introduction, class. Good morning to everyone here in Barcelona and in all different countries that are participating. First of all, I want to thank the organizers of this very nice meeting for inviting me. And I also want to thank everyone here at this moment for all their efforts they put in NFPU, because it is so important that this organization is present and helps improve lives for individuals with NF1. Today, I will talk about the genetics of neurofibromatosis type 1, genetic testing for neurofibromatosis type 1, and how it made a difference in the care for individuals with NF1. I'm not going to introduce NF1 because everybody knows what it is, but I have to introduce a little bit genetics, I think. So this is an image or a drawing of a cell of the body, and we have many different types of cells. We have more than 250 different cell types in the body, and maybe more. And those cells, they have a nucleus. As you can see here in the middle of the cell, there is a nucleus. And in the nucleus, we keep our DNA. And this DNA is present when a cell divides under the form of a chromosome, as you can see there at the top of this slide here. But this chromosome is a specific structure, and you can unfold it, and then you have a very long wire of DNA. And it is in this DNA that there are several different molecules that we call A, C, T, and G that are present in a specific sequence. And that is this sequence that contains the information for our genes. So we have about 20,000 genes that contain the code for the protein. And all these proteins that are made by the cells have a very strong effect on how the cells function and how the body functions. We know that these 20,000 genes are present in every cell of the body, but not every cell needs the code for every gene. You can compare it more or less with the book with recipes. Everybody has cooking books in the house, maybe several cooking books. We have all those recipes, but you never make all the recipes in a cooking book, right? You just choose the one that you want for that particular day or week or year or whatever. It's the same in our cell. All the cells have all the recipes for the human body, but they just take what they need to fulfill their function in that particular cell. So one of those 20,000 genes is a gene called NF1, which codes for a protein. And this gene is mutated, so it has an error. It has a different variant in individuals with NF1. It was discovered 35 years ago in 1990 by two different groups. And here you see the head of one of those two groups. This is Francis Collins, who was sitting on a haystack showing a needle, where he explained that I found the proverbial needle in the haystack. This was 1990. At that time, it was quite difficult to identify a gene. Moreover, it was a large gene if you compare it to other genes, and it contains a lot of code for a large protein, and the protein itself had 2,818 amino acids, and the gene itself has more than 8,000 letters of genetic code. At that time, it was also clear that 50% of individuals had no parent affected, and the genetic variant that was causing neurofibromatosis type 1 originated in themselves. So they could transfer it to their parents, to their offspring, but their parents were unaffected. When we looked initially in 1990 at the sequence of the NF1 gene, we didn't know anything about the biology of neurofibromatosis type 1. We saw immediately that the code contains somewhere a fragment in the protein that was formed that made it a protein that could downregulate the activity of a protein, another protein that was called RAS. And the RAS protein is very much involved in regulating cell division, cell growth, and so on. So we have this whole cascade of one protein that activates another protein, that activates yet another protein, and so on, that finally makes a cell grow. And this is called the RAS MAP kinase pathway. And it was clear that neurofibromin should function here, where it inactivated RAS once it was activated to grow, to make the cell grow and divide. So it is kind of a brake, like in a car. You have an accelerator to make the car move, and you have a brake to stop the car. And sometimes you want to move, sometimes you want to stop. The same with the cell. Sometimes the cell has to divide, and then at a certain moment, the cell has to stop dividing. And neurofibromin functions as one of the different breaks in the cell, and it breaks the growth of the cell that is induced through this RAS protein. So we knew at that moment that NF1 very much is a tumor suppressor gene, meaning that from the two copies of the NF1 gene, because we have every gene in two copies, one inherited from the father, one inherited from the mother. From those two copies, we know that one of the two copies has a variant that is associated with the neurofibromatosis type 1, but then there is still on the other chromosome a normal copy of the NF1 gene that produces a normal neurofibromin protein. So this cell very much can still downregulate RAS because one of the two breaks is still available. But if in a particular cell, the second normal copy is inactivated or is lost, then no break is anymore present to slow down the cell growth once it is stimulated. So you can accelerate with your car, but your brakes are not functioning. And then, of course, you know you will run into trouble. And this is then what happens. These cells grow more than normal, and they form tumors. And people from my lab and from other labs have been looking in all kinds of tumors that you can see in individuals with NF1 and in specific cell types of these tumors, and have been able to show that in these tumors, the normal copy of the NF1 gene is also inactivated. So in this schematic, you see that in the early embryonic life, all the cells of the embryo and of the newborn have one of the two copies of the NF1 gene inactivated because it was inherited from one of the parents, or it started with a new mutation. And then further on, there are cells that by accident also happen to have the second copy of the NF1 gene, let's say the spare copy, inactivated. And in those cells that are here marked in black, there is no neurofibromin produced. And there, let's say like neurofibromas can originate, or other tumors that are associated with neurofibromatosis type 1. But then we were very much surprised that we found the same mechanism also in non-tumoral features, such as the café au lait spot. Like a café au lait spot is just a spot, it's just hyperpigmentation. When we looked at the melanocytes in these café au lait spots, we also found that these melanocytes had a normal copy of the NF1 gene inactivated. So it follows the same principle. A second lesion that can be seen in individuals with NF1 is a bone lesion. It's the lesion here at the lower leg that is sometimes bowed at birth. It fractures after birth, sometimes before birth, and can make a false joint because it doesn't stick together anymore and it's not healing. We get a false joint, which is called pseudarthrosis in medical terminology. And we also found that at the region of the pseudarthrosis, there were cells where the spare copy of NF1, so the second copy of NF1 was inactivated, as you can see, for instance, here. Yeah. So these are benign abnormalities. They are not associated with the tumor or with anything that is with the tumoral growth, but they also have the same abnormalities as we see in the tumors. The bottom line of all these things is that there is an overactivation of RAS. And everything that RAS will activate, like the MAP kinase pathway with MEK and ERK. Yeah. So due to the light, it's not very well visible, but these are different café au lait spots. What we could see is that in every single café au lait spot, you have an independent inactivation of the spare copy of NF1. So all these spots have a second abnormality in the spare copy of NF1, but it's a different abnormality. After the identification of the NF1 gene itself, it was then, of course, possible not only to study the function of the NF1 protein neurofibromin, but it was also possible to start looking into patients and see what type of NF1 variant that they would have. And then we know it's a very large gene, so you can have many different possible variants that are spread all over the NF1 gene. And in a very large study here by Leuven Messiaen, when she was working in Birmingham in Alabama in the United States, she looked at more than 8,000 different individuals with NF1 from different families. So they were not related to each other. And in these more than 8,000 individuals, she found more than 3,200 different individual variants in the NF1 gene that were causing the neurofibromatosis type 1. So it is relatively complex to look at the mutations in the NF1 gene because they are not all clustered in one region. So it is a very complicated process analyzing them. And in most individuals, you will find another genetic variant, except for the one group of individuals. About 5% of all individuals with NF1 will have the same type of abnormality, and that's a deletion. So the absence of the NF1 gene and some surrounding genes. So it seems as part of the chromosome has been cut out and lost. And it's virtually always the same size that is lost due to some, let's say, critical genetic regions that are susceptible to breaking down and losing the material that is present there. So at this moment, it is already a lot easier to look at all the different variants because we have better genetic techniques to do the analysis, but we still at this moment need to use different techniques to detect all potential abnormalities in the NF1 gene. Now, when we look at the deletions, you can see this is a schematic of the chromosome where the NF1 gene is in the middle here of this schematic. And you have different types of deletions, but this one is the most frequent deletion in red here. And the breakpoints are usually at the same position. So it is with all these other genes here that are co-deleted. And what we saw already quite some time ago is if we compare the individuals with an NF1, what we call microdeletion, then we saw that the risk that one of their plexiforms neurofibroma would become malignant was 2 to 3 times higher than in individuals who had another genetic variant. So that was important news, and it showed already that genetic testing and the genetic analysis might have an impact at least in 5% of the individuals for their follow-up, because it was then advised that these people would have a more intensive follow-up to detect early on potential malignant degeneration. But from a research point of view, we were in my lab interested in finding out why these individuals had an increased risk. And the hypothesis was that one of those genes that was also deleted was responsible for it. And so we started to look in all these genes, and together with labs from other universities, such as Harvard University and Cardiff and from Paris, we collaborated and we found that it is this gene here, SUS12, which is responsible for the increased risk of neurofibromas, but also for the increased risk of malignancies. We found out what it in fact was doing, this SUS12 gene. We published this with all the other people that were involved. And at that time, it also became important, as you can see here, that it does something with the structure of the chromosome, that detecting this abnormal chromatin structure of the chromosome now is one of the, let's say, tests that pathologists who look under the microscope at tissue from a tumor use to find out if it is a malignant neurofibroma or not. We call it an MPNST, a malignant peripheral nerve sheath tumor. So looking into the microdeletion, finding out the gene that was important for the increased risk of malignancy led to a better surveillance of the patients, but also to a better diagnosis of malignant peripheral nerve sheath tumors under the microscope. And then, of course, we also saw patients of individuals who had a mosaic distribution, say not, the body was not completely involved with cafe-au-lait spot, but as you can see here in this lady, only the left side of the body and the left arm and the left armpit was involved. You can see here. And there we hypothesized that the mutation was of course not present in the parents, was not present at fertilization in the egg, but then during early embryonic development, one of those cells made an error by multiplying and dividing the DNA over the 2 following cells in such a way that there was a variant introduced in the NF1 gene. Then, of course, all cells that are further developed from this single cell will have the same abnormality. And that's why you can have on one side of the body or only one arm or one leg the signs of neurofibromatosis type 1. Of course, you still need in the cafe-au-lait spots to inactivate the spare copy of NF1 that we have. And by doing so, we can now look at the different cafe-au-lait spots, 1, 2, and 3. We can take a biopsy from them, isolate the melanocytes, and look at the melanocytes to find the NF1 variant that started the process. And then in every cafe-au-lait spot, of course, we find also the variant in the other NF1 gene. But if this person wants to have children, because half of the body seems to be affected, it is also possible that the oocytes on one side of the body might have the same variant, and that this person might be able to transmit this variant to the offspring, to the children. But if we look in the blood of this individual, we didn't find any variant in the NF1 gene, only in the melanocytes. So if you do this, you can obtain the NF variant that might potentially be transmitted to the next generation. And if this individual wants to have a prenatal test, then this is possible because the variant is at hand and can be detected. Because it is very difficult if you don't know upfront the identity of the NF1 variant to look at it during a pregnancy in a punction of the amniotic fluid or a chorionic villus sampling, because then you never know that you might have missed the variant because you don't know where you had to look. So therefore, it's very important that we can now offer this also to patients with mosaicism. Okay. Then, of course, there is this possibility of neurofibromatosis type 1 due to cafe-au-lait spots, but also, as Klaas has mentioned this morning, we identified another condition that looks like neurofibromatosis type 1 because of the cafe-au-lait spots. We identified variants in another gene called SPRED1, and then subsequently this condition was called Legius syndrome. What we know about this condition is that it has the same cafe-au-lait spots with or without freckling in the armpits as in NF1, but it does not have the NF1 associated tumors such as neurofibromas, plexiform neurofibromas, optic pathway gliomas, brain gliomas, and so on and so on. And it's only in a small group of children with cafe-au-lait spots that are suspected of having NF1 that you find this variant. But also, again, for this small percentage of children, it makes a big difference because they don't need any follow-up for optic pathway glioma. They don't need to be followed for neurofibromas and so on. If we look at what we know now due to the work of Leuven Messiaen, it is that we have this microdeletion. We have more neurofibromas and more risk of a malignancy. We have some 2 other variants that are associated especially with neurofibromas very close to the spine. And then there is 2.5% of variants in the NF1 gene that are also associated with cafe-au-lait spots, but not with the other NF1 associated tumors. So if you see individuals in families and they have only cafe-au-lait spots and no neurofibromas, optic pathway gliomas, no Lisch nodules, that they can have either this variant in NF1, one of those 3, or they can have Legius syndrome. So pure from the clinical point of view, you cannot make the difference, and you have to do the analysis of the genetic variant to make sure it's SPRED1 or NF1. And by building the revised diagnostic criteria a couple of years ago, we also now included an NF1 variant as one of the 2 criteria that have to be fulfilled. So we can still make a diagnosis of NF1 if you do not have access to genetic testing, because there are many other criteria that help you to make a diagnosis of NF1, but now also the molecular testing showing a variant in the NF1 gene that is pathogenic is an additional criterion. Having this in itself is not sufficient because we know that in some rare cases you might have a variant that is limited to the blood cells without any symptoms of NF1. So you need on top of that something else. We also looked at the tumors and how they evolve from benign into malignant tumors. And we identified about 15 years ago that that there is a stage in between the benign plexiform neurofibroma and the malignant peripheral nerve sheath tumors, and this is called atypical neurofibroma. It has some features of both. It is not malignant at that time, but it is already on the way of becoming malignant. So we identified in these atypical neurofibromas that the gene CDKN2A was inactivated, usually by loss of the part of the chromosome where this gene is localized. So we had an additional way of identifying these atypical neurofibromas, and we could also show that they were on the way of becoming malignant and they were pre-malignant tumors. And so in this schematic, you see that this is the nerve. In order to form a plexiform neurofibroma, you need to inactivate the spare copy of the NF1 gene, and you get a benign neurofibroma. If on top of this you will have loss of the gene CDKN2A, you will get an atypical neurofibroma. And if on top of that you have, for instance, loss of SUS12 or EED, then you are already in the malignant process. So it shows you the different steps that we know and understand at the moment, how you go from a completely benign to a malignant tumor. And that helps also in this genetic analysis on tumors, helps also decide when to do surgery. Because if you're in the pre-malignant state and you think that in the future this tumor might become malignant, then it is better to remove it. And then the last topic is pre-implantation genetic testing. This is testing of the embryo before it is implanted in the uterus, in the womb. You can do this only if you go through the process of in vitro fertilization. This procedure is not available all over the world in equal amounts. And sometimes it is reimbursed by the healthcare system, sometimes not. Sometimes only a few cycles are reimbursed, sometimes more cycles are reimbursed. So it's very different from country to country if it's available or not. But in those countries where it is available, it is a way of avoiding the birth of a child with neurofibromatosis type 1 without having to go through the painful process of testing during pregnancy, followed by pregnancy termination if you get bad news. So if an egg is taken from the ovaries of the woman, it can be fertilized with a sperm cell from the partner. And then after a couple of days, in the beginning, it was day 3. Now it is day 5 that they take a biopsy from this embryo. Then the embryo is frozen. The analysis can be performed. And then later on, well, we know there are several embryos. We know the ones that have inherited the NF1 variant and those that did not. And then one of those that did not inherit the NF1 variant is then transferred into the womb of the woman. And then we hope she is pregnant. And if she is pregnant, then we know that the NF1 variant has not been transmitted. So most centers are now going from the blastomere biopsy to what is called trophectoderm biopsy at day 5, which has a higher rate of success if you implant afterwards the embryo. So this makes a big difference because it adds another choice to a couple where one of the two partners has NF1. So genetics of NF1 allows for a proper diagnosis. Sometimes in a young child, you only have café au lait spots. None of the parents are affected. And then having the molecular diagnosis might help to prove what is going on at a very early time point in life, if it's available in the country. It's now part of the diagnostic criteria, but it is not necessary. And in some cases, genetic variant predicts future tumor risks, and you can adjust the follow-up accordingly. It gives us more insight in the origin of the potential NF1 complications, like the malignant transformation, with better follow-up of individuals with a lower risk or a higher risk of malignant tumors. And then there is this possibility of prenatal, but also now more recently pre-implantation genetic testing for couples where one of the two partners has NF1. And these are all the different people in my lab when I was still in place that have done all the work and all the external collaborations. And I thank all of you for your attention.